Hello students, welcome to lecture 2 of week 9. So in this lecture we will look at what is evolution, what are different parameters that we use to estimate evolution, the methods, and how we can determine evolution or estimate evolution through various evolutionary methods for publications. So first of all, like what is evolution? You might have read about it earlier. So basically we all have evolved from a single cell through over a period of time, okay, encompassing several billion years, and so basically slowly evolution has happened, and that has brought changes which are heritable. That means which can pass from one generation to another, okay, and those changes that are advantageous get selected, that is natural selection, right? And each species try to survive in the environment, adapt, okay, so that is the next selection, survival of the fittest, and the changes that are heritable and advantageous are taken forward, and this way anything and everything that we see around us has evolved. Many of the organisms that we see today, their ancestors are lost, okay, which are for that, like fossil records can be helpful to determine evolution in that case. And you all know that it all started with the efforts of Charles Darwin, who published a book called On the Origin of Species in 1859, and he gave various theories that changed the way people used to think about evolution. Okay, so basically the keywords in this evolutionary processes are like natural selection, okay, like how among the individuals which are the best fitted or that have some change that can lead to better adaptation are selected, and basically adaptation goes hand in hand with the natural selection, okay, and basically what we see is the phenotype, but what is happening at the back end is at the genotype, at the genome level. So these changes happen at the genome level, which are translated or expressed as a phenotype, and that are inherited further. So like in this picture you can see that basically genome started from when the earth formed, and very small organisms happened, and they gave rise to very massive bigger organisms, also reptiles and dinosaurs are now extinct, okay, because they could not survive in the changing environment. And the species that could survive and evolve, like you can see the good examples are humans, peacocks, which are very nice, beautiful long tail birds, but still despite of their beauty and long size, they could survive evolution because they adapted to the environment, okay. So and other thing that you can observe here is that a kind of timeline where we can see that which organism evolved at what point of time, which is also very important, right? And also the speciation event that lead to differences between the organisms, right, at like diversification into various species, okay. And then we can connect and anything that we see around us in the form of a evolutionary tree, where there is a root, okay, and there are clades, like this is one clade, okay, this entire thing can be also be one clade, okay, and this is one clade, and this entire thing can also be one clade, okay. So where depending upon like how you want to look at it, and this tells that, and basically in a clade the organisms are mostly related to each other, okay, like if you see in this one, these are all mammals, okay. So this way we can try to connect and build a tree of all the life forms that we see around us. This is only for the mammals, okay. Now like some examples of adaptation and how things evolved. You can see that in whale the hand in human looks something like this, in bird it is the same structure, looks like this, in dog it has taken this shape, and in human it has taken this shape. So which tells that basically how the bone structure and which are homologous earlier in tetrapods have changed to something with a very finger like structure in humans, right? Another example that Darwin also quoted, that in the case of finches, which is called Darwin's finches in the Galapagos island that he visited. So you can see that the finches have evolved or changed the shape of their beaks over a period of time, okay, which is to adapt to the environment, or okay. So this is one of the example that and they diversified into like 4 different species, you can see, okay. And like change in the like beak structure is one of the observable phenotype. So this is how they evolved. Humans is very good example, where like in primates you can see that this is human, and the other one, chimpanzee, gorilla, orangutan, gibbon, they all look like in structurally similar, but humans got the erect structure, backbone, and development of mind, facial features, and all these things evolved further in the case of Homo sapiens. And basically all our primates and all had a common single common ancestor. Okay, so basically to determine evolution, what we see, we can do morphological analysis also, but that may be misleading, because we sometimes morphologically 2 things may look similar, but they may have a very different origin, okay, or just by chance also, or the way you are looking, or you know, the parameter that you have to examine it morphologically may lead to a lot of, you know, misleading, you know, unless you are a, you know, expert, and even experts can make error morphologically, and that is why we need molecular methods. And for molecular method, DNA, RNA, proteins are the best ways to you know, estimate evolution. So these are generally used, DNA, RNA, and proteins, okay, and these are the basis of the molecular evolution, okay. And that is why like genome sequencing that we have studied earlier, sequencing of genomes, transcriptomes, provides a lot of insights, like how what we see as a feature, that what was the origin of it, or whether it has evolved in recent times and in that species, okay. So for example, here like you can say that a single DNA sequence is mutation created 3 different forms, of which one was unsustainable because it was something that was leading to a stop codon, or something that was not tolerable or adaptive to that environment, but other ones could go forward and further changed and maybe lost over after a period of time, but some could sustain, and the different versions of the initial sequence that we started with, we could see today in different species that we see around us, okay. So this is how DNA changes at the species level. And if you compare like a family such as in bacteria, a simple example, Enterobacteriaceae is a family, there could be several members, Shigella, Salmonella, E. coli. They may have several proteins which are identical, but several proteins which are species specific or have changed a lot, okay, and all of those belong to the same Enterococci family. So this is one way, like some things are retained and some things undergo changes to give species specific advantages. So we should also understand a few concepts, such as what are homologs, orthologs, and paralogs. Okay, so basically homologs are sequences that originate from a single ancestor or the same ancestor, right. So like globulin genes, or there are several other cases also that we will discuss, several examples. Orthologous gene is something that had a common origin, and now it is present in all the species that you are considering, right. So and but it may have evolved in those, and orthologous genes is also a kind of homologous gene, and but it has a like it is commonly present across all species, right. And similarly paralogous genes, so like for example there was one gene that became, okay, so now A was doing one biological function. So now if it has become 2 copies, like paralog, like that means duplications within a genome. So now this A1 copy can do the function which A was doing, but A2 is now free to do some other function, right, and it can evolve, and even if something changes in A2, the organism can still survive because it already has A1 which can do the job which was supposed to be done, because genes have certain functions that you cannot stop or discontinue or suppress immediately, right. It may happen over a period of time, but not, okay, that may not be tolerable, and that is why like paralogous genes are very important in understanding that, and giving new leading to new origin of genes and their functions. Okay, so basically it become clear from this example also. You can see that there was early globulin gene that became after gene duplication into alpha and beta chain genes. Okay, now if you see three species, frog, chick and mouse. So basically the alpha gene is present across all frog, chick and mouse. You can see alpha, 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 alpha present in all three. That means they are orthologous, commonly present in all these species, because and also descended from a common origin. That was the early globulin gene. And the other copy, the beta gene also, you can see that it is present in mouse, chick and frog. So again it is an orthologous gene, so duplicated an ortholog. Okay, and then if you see carefully, then like mouse has two copies of both alpha and beta. Okay, so and similarly like chick also. So basically those are the paralogs, okay, which are now after in a single species a gene because of like segmental duplications or other kind of whole genome duplication that happen in genomes, you know, several regions duplicate and the genes which are there in those regions also duplicate. Okay, so like in this picture also you can see that there are initially there was gene A that got you know during speciation event became gene A and gene B, okay, with slight during the when the two species diverged. Now in the two species, now they both can have A1 and A2, okay, which are basically the paralogs, okay, and the orthologs gene A and A2 both are present in both of those. Okay, similarly B is present in both of those. Okay, so these will be doing almost similar function, but there could be another gene which have originated in a separate species, okay, which is distant from it and not, you know, directly related to this species, and the gene is also doing similar function. Okay, it could be because of survival in environment or okay, so that gene was not initially you know linked to the gene A, but this gene C is unrelated but performs similar function, and that is why it will be called an analog. So doing have a separate origin but doing the same function, analogous function. Okay, so through example, if you see like these are histone proteins, histone H1.2 and H1.1, okay, in human and chimpanzee. Okay, these are the paralogs. Okay, these are the orthologs present in both human and chimpanzee, H1.1, H1.1.1. Okay, and there is an analogous protein also present in bacteria, HNS, that also do similar job. Right. So now if we see that at the molecular level, so basically this is the amino acid sequence alignment of the histone H1 proteins from residue 120 to 180. Okay, so in human, chimpanzee, mouse, cow, you can see that there are several positions like denoted by these stars which are conserved, they do not change. Okay, but there are some changes that happen, like in this case, like there is A, B, V, A, so that means some changes have happened. Okay, that means that position can be changed a bit, that amino acid, okay, but still it will, you know, because of the significance of that amino acid at that position, it can undergo like some homologous substitutions mainly, right. And in this case also you can see that's a kind of conservative change, whereas this is a non-conservative change which is at this position, right. So basically some positions will always remain which are critical will remain conserved. Some positions can allow changes. So we are looking at the amino acid level, but the change would have happened at the DNA level, right, and which have changed the amino acid. Now if the change is homologous or non-homologous, or basically you can say synonymous versus non-synonymous change. So okay, synonymous ones are more tolerable, but non-synonymous can also lead to you know, evolution if they are sustainable and give some advantage to that species. Okay, and similarly like paralogous genes are the genes that within a single genome, one gene can make multiple copies of it by various events, duplication events that happen in the genome, right? And that is why we have, like, for example, HOX-A cluster containing 13 genes, HOX-B, HOX-C. In different species, you can see, like, the same gene is having like, you can call that a gene family, having different number of members in that gene family, and some show expansion and some also show contraction. So that is also related to what advantage the genome is getting by the expansion and contraction of those genes. Then there can also be homologs, like in this case where the whole genome duplication event is creating copy of a gene, and now the new copy that has evolved also can undergo some change. Okay, so those are called homologs, right, which are paralogous genes because those are the copies of the same gene in a genome. Okay, and they have originated, but the only thing is that they have originated by a whole genome duplication event. Okay, similarly there is xenolog concept, where basically there was a speciation event that produced orthologs of a gene in two daughter species. So these are the orthologs, gene A1 and A2. Okay, so now gene A1 was present in this species, but gene A2, which was present in species 2, got horizontally transferred to species 1. Okay, and so basically it is one of the homolog of that, but that is directly transferred from one species to another, which is called the xenolog. So basically, just to summarize, evolution is a fundamental process that has shaped the life on this planet. All the things from bacteria to very large organisms around us have evolved because of evolution, and even their distinct forms you can see that, which truly you can see in the form of a the species level, genus level, family level. If you see, like, at the species, at the genus level, most of the species under it will look similar, but at the family level and when we go higher in the taxonomic tree, they will show more and more changes, and that is why how they are linked to each other and also connected through evolution. Okay, and the changes that have happened in those over the period of billions of years are adaptable changes, and that is why those changes that provided some kind of advantage like fitness, function, or survival of that organism, and those were retained, and the insights about it you can obtain by analyzing the DNA or protein sequences. That some of the method we will see later. And so basically through the evolution analysis, you can know that how the species that you observe around you are related to each other, how a particular function that is of your focus evolved. So you can do the evolution analysis at the single protein level or at the whole genome level, and construct trees to and it is not true that the tree at the protein level, at the genome level, will show similar type of shape and structure. It may show differences based on like specific evolution that has happened in that protein or DNA sequence. And through this we can also understand the concepts of natural selection, adaptation, that has happened over a period of time. And as I mentioned, like genome and transcriptome sequencing are the best ways to first get the data that you will use for the evolution analysis. Okay, so basically evolution analysis is a multifaceted area, and each of the area is you can do, like, for a new genome, all the things that are mentioned. For example, here the demographic history, genome collinearity, like how two genomes are collinear, what time species diverge, and what was the exact what things actually diverge at that point of time, the divergence between the nucleotides and the positive selection. Some genes are positive selected, there could be some unique amino substitution that provides specific advantage to that organism. Okay, some gene families are more evolved in terms of their size and structure, right? So all these we can do. Okay, and for this you also need the annotation of the genes, because you have to understand the function also when you are dealing with the evolution analysis. So you can use KEGG, AGNOG, and GEO and other kind of functional tools. right, for annotating the genes. Okay, so it's all start with, first of all you will sequence the genome, predict the genes, annotate the genes by various methods, do comparative analysis to do further annotation if needed, and other methods of annotation that we also saw earlier, right. And then we will go for the various typical evolution analysis to estimate the evolutionary things that have happened in that species. Okay, so orthologs are very important in this. So there could be genes which are, so orthologs basically, as we have seen earlier also, are the genes which have commonly present across the species that you are observing because they had a similar origin. But now in the different species they might have undergone some changes because of the speciation events or species specific changes. Like human and mouse, you can see like orthogroups, orthologs, okay, human chimpanzee, okay, mouse chimpanzee. So these are the genes which are commonly present across these. Okay, so it is not that all the genes in human I can find orthologs in chimpanzee, right. So chimpanzee and gorilla may be the closest, so I can find most of the genes as orthologs. But as I go further and further and include more and more species from like far groups in terms of the phylogenetic tree or like at the order level or family level or genus level. Okay, so so that is why we have to include only those which are commonly present across all the species that you are considering for the evolution analysis, right. So so it's a kind of trade-off that if you include more and more distant species, your ortholog number will be smaller. Okay, and okay, so you can do a far comparison but by limited number of features that are available. And if you include closer species, you will include more genes, but the information that you are going to get about the evolution will be limited to that group of organisms. Okay, so as I mentioned, there are several aspects of genomic analysis. You can do the demographic history, like at what time their population changes in terms of the population structure of an organism, right. Because population do face declines during various time periods during evolution. Okay, then we have to certainly predict the coding genes and the non-coding genes after the genome assembly is done. And then we can do genome collinearity analysis by the closer related genomes that we have, which is part of the comparative evolution analysis. We can determine the adaptive evolution, okay, and also determine the multiple signs of adaptation using various parameters such as positive selection, unique amino acid substitution, higher nucleotide divergence, gene family evolution. So we will be discussing all these 4 methods later, and use all this information by as genome wide or for a particular set of genes or for orthologs by constructing a phylogenetic tree of the species that we are doing the comparative analysis. Okay, so basically since evolution is a branching process where like populations have changed over a period of time, so it is possible logically to connect those populations into the same branches and construct the phylogenetic tree, okay, which can be of various shapes that it could be more artistic or more standard, right. And we can certainly analyze the genome that we are around us and for which good quality data is available at various, because it is not that all the genomes have been sequenced, but the common ones or you can say that the more important ones or right, you can find some data or a lot of data for those genomes, but there will be several which are missing and because they got lost during evolution and extinct or now no more existing, like mammoths for example. So those you cannot find, but you can find some fossil records if possible, or you can make assumption and hypothesis in that tree regarding those nodes and clades. So basically phylogenetics is very important here because it is a study that tries to relate the various species around us in the form of a tree using the molecular sequencing data. You can also do by morphological method, but that may not give the true tree or the correct tree. And even in the molecular method also, there are several methods, some of which can promise better or you can say more reliable tree than the others. Okay. And basically the origin of this word phylogenetics is by two words, phyle and basically phylon, basically that we call, which is mainly referring to a tribe, and the other word is like geneticos or phylogenetics, okay, which means the origin or genesis, right. So basically genesis of a phylum or a group, right, or a tribe. Okay, so basically by phylogenetic analysis we can try to relate the species around us in the form of a phylogenetic tree, and you can see this is one tree. Okay, so this is the root, okay, and this is the branching point, okay, and so the C, D, H, I, F, these are the leaves, you can say, and these are the present day organisms that you can observe, right, and this is the nodes. Okay, these are the various and this could be one clade, okay, this could be another clade. So these organisms are more related to each other in this clade, whereas these ones are also related, and these two are separated by some they are separated, but they could also be related to each other if you make a little bigger tree, right, but they may not fall into the same clade, but they could be more related that way. Okay. So basically considering just a few species will give you only some information or partial information, and that is why considering more and more species will actually give the correct shape, structure, and the location of a particular species in that tree. Okay, so basically it is called a tree because it has root, branches, nodes, leaves, which you can also see in a tree, that that is why it is called a tree, right. And DNA and protein sequences are generally used to compare, and there are also, like we will also see in later parts, both have their own advantages and disadvantages. Like many things in many analysis you would like to go for proteins sequences if you are considering very distant sequence, because at least at the protein level there you can see some homology, whereas in the case of DNA you would like to use DNA sequences in case of very closely related genomes. Like if I do only the primate analysis, I may consider DNA sequences, whereas if I want to compare human, spinach, frog, and bacteria, protein level analysis will or multiple sequence alignment will give me at least something. Okay, so basically if we see the overall method how we go about a phylogenetic analysis, first of all we should have the genome in hand or the sequences that you are interested in based on the hypothesis and your research problem. So those sequences you will find in the public databases. You should download them into the same format for each species. Again, like the quality of data and the input sequence is very important, so you should use Swissprot or EBI, EMBL, or like but if that is not available, you can also use the NCBI, RefSeq, and even an NR database. Okay, so the point here is that better the data, input data, you should also do the manual curation to further check. For example, somebody has included or mislabeled a sequence, that becomes a part of your analysis and will lead to misleading results. So you should be careful there. Okay, so once you have done the downloaded the sequences, done the data curation, then you will do the alignment. And since you are dealing with multiple sequences, you are dealing with multiple sequence alignment. So you'll align them, and in the multiple sequence alignment, since you are dealing with multiple sequences, cluster W may be little slow, but that also is acceptable. But like MAFFT is one of the method for the multiple sequence alignment. So after the alignment is done, you can use that alignment or use that as that element as an input to another program, okay, which can do the which can help you to construct the phylogenetic tree based on any evolutionary model. For example, maximum likelihood is one of the model that is preferred. Okay, Then you will use a software to construct the tree such as RaxML or Phylip or etc. Bootstrapping is done because bootstrapping means iterations, that how, like using subsets or subsampling, you will repeat the analysis so that you can confirm that the species that you observed at a particular point or position in a tree is appearing at the same point anytime or anyway. I am repeating that calculation, right? So that will increase the confidence in the tree that you have built regarding the structure and the position of various species in that tree, right? So that is why bootstrapping is very important, and like 100 is a good number for bootstrapping. And then you will visualize that tree. There are several tools to visualize the tree. Fig tree is one, and basically you can pick something which is more cartoonic or a standard one based on like where and how you want to report the result. Okay. So the two methods which are commonly used are the character based methods and distance based methods. So distance based methods are more simpler and more true for, I would say, that the closely related species, but and then comes the character based methods, which like maximum parsimony, that considers only a few positions that are important, right, and does not include all the positions of a sequence or the alignment that you have. Whereas the maximum likelihood will calculate the log sort ratio for each and every position and calculate the probability, at each, and so basically it will be considering each and every character in the alignment, and that is why it will be slower and but will be more accurate. Okay, so basically the maximum parsimony, maximum likelihood and distance based methods like neighbor joining etc. are the commonly used methods for phylogenetic constructions. So we will be having a separate lecture for taxonomy and phylogeny, and there I will be going to details of these. So now we should move on to like the other after the tree, and we have the sequences sequencing done and multiple sequence alignment done, the orthology determined. So we will go for the analysis. So look for the signs of adaptations or adaptive evolution. Okay, so there could be several ways of doing this. So for example, higher rates of evolution. Okay, there will always be some positions in the sequence which are more conserved and do not change because they are critical for the function, but there are some positions which are are changing, evolving fast, and they could be more responsible for the species specific advantages and are maybe a good indicators of the evolution. So we have to determine those positions. Then there could be some unique amino acid substitutions that, so amino acid substitutions do happen because the DNA undergo mutation by several ways of which are other processes, right? And but due to that changes at the DNA level, the protein sequence, if that happens at the coding region, also change. Now that change could lead to a functional impact if that is going to change the amino acid, and in such a way that because at any position in a protein, the amino acid shape, its structure, its charge, right, everything matters. Okay, so if that change is synonymous, okay, it may not matter much, but if it is non-synonymous or leading to some kind of functional impact, so those changes are more important. And positive selection, because some genes are under the positive selection, and we will see that how to determine those genes also. And so for a particular genome, there could be some regions which are highly evolving, or some genes having unique amino substitutions, or some genes under positive selection. So you can consider at least like more than one, or basically two or more of such signatures to say that okay, the gene is showing multiple signs of adaptation. Okay, so this is multiple signs of adaptation. Okay, so high rate of evolution, because as I mentioned, there are certain regions or genomic regions that are more prone or basically are undergoing evolution or changes over a period of time than the rest of the genome, and these could be because of changes, or basically response to the environment, or making the species more fitter and better in that environment. Okay, and also those changes are heritable, and what you see in these species or different individuals of that species, you could find that at that position that change is there in all the individuals of that species. So those are the molecular markers of adaptive evolution, and they are under less selection pressure, okay, because they do not be a part of the critical functions such as the enzymatic site or the position that lead that are important for the protein folding. Okay, so we have to determine these positions which are under the higher rate of evolution. Okay. So for this, basically the root to tip distance is so once you build the tree, you can see that for a particular species or the two species that you are considering, they are coming together in that clade, but one is showing a little longer branch length compared to another one. So from the root to that species, there could be a longer root to tip distance. Tip is the actual organism or the leaf that you are considering in that tree, right? So that means among the two species which are coming next to each other, one species is actually like the proteins or genes that you are considering for building that tree, or the set of genes that you have considered for that tree, okay, have a higher root to tip branch length. So that means root to tip distance. So that means those are having the higher rates of evolution in those sequences. So basically there is a package, R package Philo, that we have also seen earlier when we discussed the R basically basics, and there I also mentioned some of the packages that you can routinely use in genomic analysis. So Philo we discussed, Philo we discussed at that point. Okay, so that can be used to compute the root to tip branch length. Okay, and this could be a very good indicator of the regions or genes which are having higher rates of evolution. Another important aspect is the positions which are showing the unique amino acid substitution, and that, so basically amino acid substitutions will be there, but it could be unique because they are occurring only in the species of your interest, not in the other ones, and also that change that has happened is also giving rise to some kind of functional impact. So it is not a change without an impact, but a change which is leading to some kind of positive impact. Okay, so those kind of mutations that happen in a protein are determined using the unique amino substitutions, and those amino substitutions also tell that basically those might have happened to make the organism more adaptable to the environment, for the emergence of novel traits, or to be, and basically this is the basis of the diversity that we observe on this planet. Okay, so for example, in this slide you can see that there are 5 species. So the proteins of these 5 species were aligned, and you can see that this region was conserved across all, but in the species of your interest there was one change at this position, whereas all the other species were showing the same amino acid, which means that this is a species specific or a unique amino acid substitution that has happened. And then there are software such as SIFT which can determine whether that change which has happened is leading to some kind of significant functional impact also. Okay, so basically to note here is that you should have the multiple sequence alignment, before that the data or the sequences to compare, and then do the multiple sequence alignment, and then you can observe these kind of changes. Okay, so to identify the genes with unique amino substitutions, and that also are relevant for the functional impact. So there are like sorting intolerance from tolerance analysis or SIFT analysis, which can predict the changes which are leading to some kind of a functional impact. Okay, so SIFT will give score to each and every variant, and based on those scores you can determine that what positions or amino substitutions lead to some kind of change or the functional impact, and others you may not consider for your analysis. Okay, then positive selection is another very important aspect of evolution analysis, that because there are some genes which are positively selected and some which are that may not be under positive selection. Okay, so those genes can provide insights into what functions, because genes do functions, and if the gene is positive selected, that means that function is also positive selected and evolved, and that means that function is more important and relevant for the survival of that species. Okay, and that is why it was positive selected and also evolved. Okay, so for this you have to do the dn by ds, and that means the non-synonymous versus synonymous amino acid mutation. So we have seen mutations happen in proteins because of the change in the DNA, which leads to the change in the amino acid. So that change could be synonymous or non-synonymous. That means synonymous means that it is not actually changing the amino acid. So basically there are, in a codon there are 3 positions. Okay, so generally if the change has happened at the 3rd position, it does not lead to change in the amino acid, whereas if the change happened as 1st or 2nd position, it may alter the amino acid. So that could be non-synonymous change. And then basically if the ratio of that synonymous to non-synonymous, or basically the non-synonymous to synonymous, dn by ds, okay, is more than 1, you can say that the gene is under positive selection. Okay, so you can see here that at the DNA level, basically this was one change, G to C. Okay, that changed the amino acid from D to R. This happened at the 1st position in this codon. Then the 2nd codon CCC was same, gave rise to P. And in this codon you can see that AT and CTA, that means again at the 1st position A changed to C, right, and it changed the amino acid to I to L. Okay, then so this is the non-synonymous change, whereas the synonymous substitution, you can see here CAA to CAG, last position amino acid, the DNA codon got changed at the 3rd position. So but did not lead to a change in the amino acid. Q was present in both. Similarly here C by A, and basically S was present in both. So basically these are the synonymous substitution that did not change the amino acid even though there was a change in the DNA sequence or in the codon. So we calculate the ratio of these. So basically if the ratio is more than 1, the gene could be under the positive selection. If the ratio is less than 1, the gene is under the purifying selection. Otherwise, if the ratio is 1, it is the neutral selection, or basically no selection scenario. Okay, so basically for this again you have to align the genes using MAFT or cluster-W. Then there is a program CodeML which can do the use the branch side model, and also available in Phylip, right? Use the Phylip. So basically you have to import the sequences in the Phylip format or any other compatible format for that program. Okay, and then you can carry out the dn by ds analysis and identify the genes that are undergoing the positive selection. Okay, in the last, basically it is also important to see, because we have seen the concepts of homologous genes, paralogs, and right. So basically for a particular gene which is involved in a function or performing a function, okay, the number of copies of their genes also matter. Okay, if that function is more required, it may have more copies. Okay, so that means the gene family, if we consider for that gene, will be bigger in size, right? Whereas if the gene, so basically if we observe that gene family has expanded, that means those families could be, or that functions maybe are more needed. Whereas if the gene family has contracted, okay, over a period of time during evolution basically, so you can say that those functions were not so important. And this we cannot do for very distant species, but by looking at the closer related species, including an outgroup and other things that we do while constructing that tree, we can observe that for example, like considering an example, like this is a tree, we are considering like this species A and B. So species A, for a gene family or that particular gene, the copy number or the number of genes present for that type of gene or that function are 20 in species A and 15 in B. For another gene or gene family, there are 20 members in species A, 25 in B. So, expansion, okay, contraction and no change between the for gene 3. So, that means no evolution. So, we can determine that gene 1 got expanded, gene family got expanded for in case of the first type of function, and there was a contraction in the second species, and this gives a very good, so basically you can do this by a cafe analysis. Cafe is a tool, and basically this can tell that how or what kind of functions or how many functions got expanded or contracted. So, like in this case, for example, this is a banyan tree, Ficus bengalensis, and Ficus religiosa is the peepal tree. Both are ficus species. You can see that this is the calculation for the gene expansion and contraction, and since the two species are closely related, you can see almost similar number of expansion contraction. Okay, green determines the plus sign with some number is the number of genes that showed expansion or gene families that show expansion, and the other one is the number of gene families that did not show basically showed a contraction. And generally the gene families that showed more than increase of 10, more than 10 are considered as highly expanded, okay, during the comparative analysis. And basically we have already discussed the significance of this, and this can really tell that what functions are got expanded, and then you can imply that those functions could be more important for the functioning of that species or for the survival, or basically giving some kind of advantage to that organism to survive in that environment. Okay, so in this lecture we covered various aspects of the evolution and evolution analysis. We looked at the concepts of homologs, paralogs, xenologs, onologs, orthologs, right, and the significance of evolution analysis and why we should do this, and what are the basic methods including the alignment and followed by the various types of evolution analysis, such as looking for the multiple signs of adaptation which comes from the positive selection, unique amino substitutions, or root to tip length, and other kind of analysis. And also in the end we looked at how the gene family expansion contraction analysis can also help you to identify or to find some insights on the function that evolved or expanded over a period of time. So, this is good for today. Thank you, and we will see you in the another class. Thank you.